right? Split plot design. Split plot design. This is another type of design. Actually, we have learned factorial design. Normally, we do factorial design, right? Let's say we have factor A, factor B as an example. Then we learn nested design. Again, we can say factor A, factor B. Then today we are going to start with split plot design. Again, we can talk about the same combination, factor A and factor B. There's a difference between these three. The first one we said, here we have factorial combination A0, A1, B0 and B1. Here also we have combination. We have already seen the difference between these two. Because in factorial, all the combinations are possible. Right? A0, A0, B1, A1, B1, B0, A1, B1. So this is the combination, all the possible combination in factorial experiment. In the nested design, yesterday we have seen all the combinations are not possible. Because here A0, B0, sorry, A0, A1, or whatever it is. Or I will go it as different terms. A1, A2, B1, B2. So A1 is not same as B1. So that we cannot produce the all possible combination. Right? So that is the problem in nested. The split plot, today we will see, we can make all the possible combination, but the precision of testing will be different. Precision of testing will be different. Actually, these two designs, that is on nested and the split plot design, these two designs are actually these two designs can be called as modification of factorial experiments. So actually, when you have possibility, you'll have to first choose the factorial experiment. If you are unable to conduct using factorial experiment, then you'll have to think about going for nested design or split plot design. So your first choice should be factorial design, factorial experiment. Right? Then if you don't have opportunity to do factorial experiment, then you go for nested or split plot. Because these nested and split plot have been treated as inferior to the factorial setup. In terms of superior, factorial is much more superior. The others are modifications of factorial, where the precision might vary. Today we will see that we have seen one precision already. What is that? For example, in nested, in our just finished example, we set area, we set site, right? When you get the comparison of area, or when you test area and site, did we use same denominator? Did we use same denominator in the?
No, sir.
No. Tell me, in area, what is the denominator that we have used?
Degree of freedom of sight.
Illa. What is the denominator? Ennathale pirichana?
Mean square of A next
Sorry,
B
mean
next
square of
We'll have to understand the question very. So we divided by, right, mean square of B, right? So this is not the real error term. It is an adjusted error term. So you see, in the second case, we have divided by mean square error. This is fine. So here, we cannot substitute that for the error term. You see, even the comparison of these two are different. Isn't it? So we didn't treat these two in an equal. We didn't look at the effect of area and the site in a same manner. Because I have some problem, right? Problem in terms of the randomization, right? Don't worry about that in detail, but from the itself, from the denominator factor, is different from area to site. In both cases, the denominators are different. It's similar. It's something like, let's say, you are having a quiz now, right? In your class, there are 80 people. 70 people sat for the quiz and got the marks. 10 people couldn't sit for the quiz. Usually, what is happening? If they don't sit for the quiz, Tanas Kumar, adutha step enna?
Special quiz.
repeat quiz.
Special quiz. For the next 10 people, you have the special quiz. Do you think the paper will be same for the special quiz?
No, no, sir.
Not same. They will be slightly different, so that they will get the marks. Now, comparing these two marks is difficult. Why?
Because both questions are different.
Both papers are different. Set of both papers are really different. That is why in A level, you have different new syllabus, old syllabus. They have been treated different, new syllabus and old syllabus, you see. So we cannot judge here. In this case, we cannot judge. Maybe this paper may be tough. The special quiz may be tough, difficult, more difficult. Isn't it? Sometimes,
Yes,
sometimes more difficult. Why? Because already in the first quiz or the first set of paper, we have taken all the information. Now we'll have to take, not to take all the information which is coming in the first paper, other than that. So that very difficult to set up paper, isn't it? So you see, sometimes the second quiz may be tough. So we cannot compare these two, actually. In real terms, we cannot compare these two group of marks. Similar to that, here, what we do is, we basically say, we want to calculate the F values. Here also we calculate the F values. Do you think these two calculated F values are in a similar condition? No, it is not similar condition. Here the F value is slightly different from this, because here the denominator factor, if you look at this, denominator factor is very, very large. The denominator factor is very large. Because the reason is the variability here in the numerator, that is also very large. So that is why very large variability can be divided by very large denominator. So that is the concept. Okay. So here, when you talk about the precision, if you talk about the precision, we have a bit of problem in the precision, right? So this is not equal precision. We can say not having equal precision. Not having equal precision. You see, they are having different precision. They are not having equal precision. One is high precise, the other one is less precise. So we should avoid, because here we have factor A, factor B in nested design. So A with less precision, B with high precision, for example, in this case. So A is tested with less precision, B is tested with high precision in nested design. But in factorial, look at these two factors, A and B. What do you think? Equal precision. A is treated similarly, B is also treated similarly. A is treated similarly, B is also treated similarly. So that you see, this is the problem in nested design. So that usually we shouldn't go for nested design unless we have problems getting all the combinations, getting some practical problems when you do the experiment, then we go for nested design. Right. Similarly, here, today we are going to start with the split plot design. Right. This is also a kind of modified form of factorial experiments. Modified form of factorial experiments. Here also, we could see later on, the precision among the factors will differ on their estimations. Right? Is it clear? Right.
Yes, sir.
Yeah. So it's like, for example, another example I will give you. I'm giving a race between two of you. This is our faculty at Kilnochi, right? So this is the A9 road. So can you remember, once you come here, there's a roundabout, right? So you can go to the faculty in both ways. Can you remember? There are ways like this. Can you remember? Yes. This is, you have the big round here. It's something like, there are two persons, let's say A and B. You are giving a race to this A and B. Starting from here, A goes through the faculty through this pathway. B is going through faculty this pathway. Who can reach the faculty? Both are walking. Both are walking. Who can reach the faculty quickly? Shortcut. You all have used this pathway. That is why I'm asking. Haven't you visited Aravial Nagar? there's a garment here, for example, there's a garment, right? So who can reach the faculty dean's office very quickly?
Yes.
A can reach because this is shorter distance. If you look at the length of the distance, right? A is shorter. So A is more precise than B here. Similar to that. Huh? Because if you come to master level, we can teach more mathematics and so on to show you why it is different, right? Now it's too much for you all to learn. That is why. Is it clear? Right. So in factorial, both distances are same. In factorial, both distances are there. In nested and split plot, it's different. Right. Now we'll come to the split plot design. So from the name itself, you can realize. Split. What do you mean by split? In Tamil and Sinhala, what do you mean by split?
Separate.
Separate. We can say further divide. In another term, we can say further divide. So what we are going to do is, normally this split plot design is applied in a situation where, let's say, one factor need large plot, another factor can be used in small plot. You see, earlier in a factorial experiment, can you remember? Let's say factor A has 2 levels, factor B has 2 levels, A0, A1, B0, B1, we have already seen, right? If you go for RCBD, I will give one setup, A0, B0, A0, B1, A1, B0, A1, B1. You see here, the size of the plot for all the combination, is it same or different? Size of the plot for all combination is it same or different?
Same.
Same for factorial experiment. For factorial experiment, this is true. But in split plot design, which is not true. You see, I'm going to talk about the split plot design. I'm just giving initial information, then we will go back. So it should be equal, huh? Assume this are equal. Now, here, let's say I have factor A. Let's say A1, A2, A3. So you see A1, A2, A3. So the whole plot is belongs to A1. Right. Let's say we have factor B. I will give an example later on. Factor B, again, let's say B1, B2, B3. Right? Now, this is actually B1, B2, B3. B1, B2, B3. Before randomization, B1, B2, B3. Right. Now tell me, what is A1? How big is the A1 plot? Plot of the A1. Plot of the A1. Plot of the A1 is this. The whole area is A1. Right? I will say this is the A1. Right? Now tell me, what is the plot of B1? Plot of B1 or B1 or B2 or B3, all are same. So this is the plot of B1. What about the third one? We'll use another color. What is the plot of the combination A1, B1, for example? Oh, I will say A3 and B1, huh? Because I can color it out. A3 and B1. Show me. What is A3, B1? This area. So A3 and B1 is that area. Now, tell me, how many plot sizes are there? For A1, A2, A3, and B1, B2, B3. Are the plot sizes are same? For A1, A2, 3, A3. What is the plot size? How big the plot size? Which one is bigger? A1 plot size or B1 plot size?
A1.
A1. So A1 is called as large plot. A1 is called as a large plot. So it is also called as main plot. So these are the names that I'm talking about. Large plot. What about B1? Small plot. Or we can say subplot. What is the difference between B1 size plot and the combination of A and B? Are they the same or different? Same, isn't it? B1 and the combination of A and B are same, so small plot. So how many plot sizes are there here? Anyone?
Two.
Two plot size. So one is a large plot. The other one is called as split plot. Right? Yeah. Only two plot sizes are there. So A is placed in main plot, B is placed in subplot. So this is the concept of split plot. Right? We will come to this a little later. Any questions? We can do some lectures, then we will come back. Give me a second. I'm going to talk about split plot design, some theory part, right? I gave you the concept. Right? We are having more than one plot size. That is the concept. We will see why we have more than one plot size. Right? Right. Usually, we have factorial setup. All these combinations are there. A1, A0, A1 combination with B0, B1 as an example. All combinations are there. When the assignments of treatments at random can cause difficulties. So because of that, we go for split plot design. Right? Large scale machinery required for one factor, but not for other factor. So sometimes we spoke about two plot size. One is very big, right? We said this is the main plot. I will say this is main plot. Then we divide this into smaller one. This is subplot. Why some factor, main plot, factor A is needed the main plot or the large plot. Factor B needed the subplot. What is the reason behind? Why in practical that we need a different plot size? For example, look at it. Irrigation and tillage. I'm doing tillage using tractor. I'm using tillage in tractor. My experiment is based on tillage and irrigation. I will take tillage first. Tillage. I'm going to do the tillage. Tillage can be done by what method? Anyone? I'm going to ask question, agriculture question. Tilini Rajapaksa, tillage kiyane monada? If I don't hear anything, I will remove. Huh?
Sir,
Yeah, tillage kiyane monada? You can answer in any language. Huh? Tilini.
sir, pasa burul karana, loose.
Yeah. Okay, we are loosening the soil. Fine. What are the techniques that we are using? Koma karana pula. Mona the kramayak tin. Mona kramayakin api karana pula. Mona bhavita kalla apita karana pula. What are the things that we can use?
Plow.
Plow. Plow. What type of plow?
Disc plow.
Yeah. Disc plow mean mounted to what? Disc plow කියන්නේ අතින් ගිහලා මේව හරන්න පුළුවන්. ඩිස්ක් එකක් අරන් ගිහලා. තිලිනි? ඩිස්ක් plow කොකටද මවුන්ට් කරන්නේ අපි? අස්සයට සාමාන්‍යයෙන් දැකලා නැද්ද? Tarshani, tell me, disc plow mounted to what equipment or instrument or animal? Huh? Huh?
Tractor.
Tractor, yes. Tractor mounted. Maybe two-wheel or usually disc plow, not really two-wheel, it is four-wheel tractor. Two-wheel tractor, moldboard usually. Plow is moldboard, not disc plow usually. Right. So now, look at. Now I am going to use tractor for tillage, because tillage is one type of experiment for my research. Tillage, there are different, using tractor, four-wheel tractor, using tractor two-wheel, using let's say buffalo, right? Using, what are the other, bull? Usually we don't use very rare, buffalo, bull. These are my 4 treatments. Factor A, tillage is factor A. Factor B is variety, right? Factor B is variety, paddy variety. Let's say BG 350, some variety, doesn't matter. 3 varieties. I have 3 varieties, 3 or 4 varieties, doesn't matter. Now, tell me, can we have combination? This is 2 meter. This is 2 meter. Can we have combination of, let's say, this is, I say A1, A2, A3, A4. This is B1, B2, B3. Now tell me, can I have combination of A1 and B1 in this plot? Anyone? This plot size is 2 meter, 2 into 2 meter. Can I have a treatment? What is this treatment? A1, B1. What is this combination? A1 and B1.
Four-wheel tractor tillage in BG 350.
Okay.
It is
Good.
impossible.
Is it possible to have a plot 2 into 2 meter to combine these 2 levels of factors?
No,
No. Why?
because
Why
tractor size is bigger than that.
Tractor is because tractor need, if you park the tractor, it may need this way roughly 3 meter, at least 2 meter, just to park the tractor. Now tractor has to move and do the tillage, so we need a large plot. Let's say this is 10 meter as an example. So this is fairly good for tractor to move, at least this is the minimum, huh? So this is a tractor need a large plot, you see. But variety, does it need a large plot? No, variety we can plant in 2 into 2 meter, for example. In 2 into 2 meter, how many paddy plants you can plant? What is the spacing? Danesh Kumar? If it is transplanted, what is the plant density? 2 into 2 meter.
A bit between 2 plants, 5 centimeters.
Huh? I didn't get it. Between 2 plants?
5 centimeters.
5 centimeters. Within 2 plants? Who are the people? Agronomy? Who are the people? Agronomy? Studying agronomy?
10 into 10 centimeters.
Oh yeah, 10 into 10, slightly low, but okay. 15 into 5 centimeter or 15 into 10 centimeter, fine. So roughly, roughly how many plants do you get in 2 into 2 meter width and length?
We
Let's
have
say,
to divide 200 into 200 by
Yeah.
divided by 15 into 5 cent.
yeah, all over. So you will get huge number of plants. So more than 200 plants you can plant it. 200 paddy plants can be planted, isn't it? So for variety, small plot is okay. For variety, small plot is okay. But for tillage, so small plot that doesn't work. That is one example. Second example, irrigation. I just spoke about example, right? Irrigation. Irrigation need a large plot. Why? Example, I take one irrigation method as one irrigation method. We have different irrigation, right? Sprinkler, then a drip, basin, or whatever it is. Huh? I'll take. So you see, if it is 2 into 2 meter, can I fix a sprinkler irrigation in 2 into 2 meter? Will it possible? Sprinkler irrigation need large area, isn't it? As an example. All right. So it need large area, not small area. Sprinkler cannot practice in a small area because it is go as a circle. So this kind of factors need large plot. So that is why we have different plot size. Some factor need small plot size. Some factor need large plot size. Can you see the screen?
Yes, sir.
Right. Irrigation tillage kind of factors need a large plot size. So that we need to go for large. But plot that receive the same treatment must be grouped together. Right? For a treatment such as planting date is one treatment. We will see the example later. It may be necessary to group the treatments to facilitate the field operations. Planting date is one treatment or one factor. So planting date, every 2 weeks is the planting date. That is my experiment. So that we need to group them. So that is another reason why we go for split plot. Not necessary to have irrigation treatment or tillage treatment, but the planting dates, we can plant at once 3 varieties. For example, let's say planting dates and variety. So I have factors. Factor A, planting dates. Let's say I have 3 planting dates, right? They are an experiment. 1st of October, 15th of October, and the 30th of October. So these are 3 levels of planting dates. Let's say this is A1, A2, A3. Right? Similarly, B, we can take varieties. Let's say I have 3 varieties, V1, V2, V3. Now, if you put up the set, so this is the planting date, A1, A2, A3. This is before randomization. I will talk about randomization later. Now, we can say three variety, V1, V2, V3. V1, V2, V3. This is before randomization. Huh? This is planting on 1st of October. This is planting on 15th of October. This is planting on 30th of October. You see? We'll talk about the replication, randomization and replication little later. So this is the setup. So the levels of factor A were planted in the big or the large plot. That is the large plot, right? Levels of factor A in large plots. Levels of factor B in a small plot. Levels of factor B in a small plot. So this is the large plot concept and the small plot concept. You can read, you can take the notes a bit in a growth chamber. For example, in a growth chamber, maybe if you have visited your agronomy department, now they have the growth chambers, JICA funded growth chambers. So in the growth chambers, you have cabins. Cabin 1, cabin 2, cabin 3. So within the cabin, you can maintain the moisture, relative humidity, temperature, and so on. There is another cabin, second cabin, third cabin. Likewise, I think 6 cabins are there, 5 or 6 cabins. You can maintain the temperature and the different parameters. You can change the different climatic parameters in all 5 or 6 chambers. But within the chambers, you cannot do anything, right? For example, within the chambers, I set a temperature 30 degrees centigrade and RH is, let's say, 60% or 70%, whatever it is, 30 degrees centigrade. So in 30 degrees centigrade, for example, you can have different seeds, for example, varieties, right? Varieties, different varieties, 3 or 4 varieties. Then in the second chamber, you can have another temperature, set it. Then you can have another set of the same group of varieties. So likewise, you can do it in the laboratory also, but still it is considered as split plot concept, right? It is considered as split plot concept. So in a growth chamber experiment, some treatment must be applied to the whole chamber where the light, humidity, temperature, everything kept similar. So split plot does not need to do it always at the field level. We can do it in the laboratory also, in the chamber, right? As an example. Right, split plot, different size requirements. The split plot is a design which allows the levels of one factor to be applied to the large plots, while the levels of another factor are applied to the small plots. So this is what I have already explained. So you apply one levels of one factor into the large plot, levels of the second factor in the small plots. Large plots are whole plots or main plots that we call it as. Small plots are split plots or call it as subplots. These are some additional information. Right, now we come to the randomization. Let us talk about randomization and replication in split plot. Right, look at the randomization process. This is the importance in randomization process. The levels of the whole plot factor are randomly assigned to the main plots. So first we do the randomization, then it's like in the RCBD, using the different randomization for each block separately. I will speak about levels of the subplots are randomly assigned within each main plots using the separate randomization for each main plot. right? Levels of subplots are randomly assigned within each main plots using a separate randomization for each main plot. Right. Now, before I talk about randomization, before I give the randomization process using the slide, now I will give the randomization. I have, let's say, factor A, we have 3, A1, A2, A3. Factor B, we have again 3, let's say B1, B2, B3 as an example, right? Now I have one setup here. In randomization process, first step, randomize levels of A within, randomize the levels of A in the main plot. That is my first step. Randomize the levels of A in the main plots. For example, here I am randomized A3, A1, A2. So this is I have randomized. Right? I have randomized. That is the first step. Second step, randomize the levels of B within each main plot separately. You randomize each levels of B within each main plot separately. For example, first main plot consists of A3, so that I randomize B1, B2, B3 here. I B3, B2, B1, B2. For example, here the second. This is the first randomization. Do it separately. Then the second randomization, I say B2, B3, B1, for example. The other randomization, for example, like this. So separate randomization. So each of these main plots will be randomized by the levels of factor B. So this is called as randomization process. Right. I will give you a randomization process. Look at very carefully. I have stage treatments. These are the main plots, 3 plots, main plots. Varieties are the subplots. You see, this is called as block 1. Under the block 1, the dark color blue shaded border lines are main plots. So here we have 3 main plots. This is the first main plot, second main plot, third main plot, as an example. Right? So these are subplots. Right? Right. Look at here. Block 2. So I was talking about block 1 only. Block 2 is for replication. You need a replication of the combinations. So we need replication. You see, I have randomized the treatment first. That means the fact of A first, right? T1, T2, T3 in the main plots. Again, you randomize for the second block separately. So they are not identical. The randomization has to be done separately. Then you randomize each variety, which is subplot, within the main plot separately. This is the first randomization. Look at the second randomization, third randomization. Likewise, you randomize this. Right, you make a note of it. I will give a 2 minutes break to get a glass of water. Right, shall we continue? Hello.
Yes, sir.
Okay, right. So this is how the randomization process take place. You see, we'll have to have, this is one block. We call it as block 1. We need at least 2 blocks. Then only we can replicate. For example, here this is T3, V3. T3, V3. So within single block, do you find T3, V3 anywhere else? Is it replicated? Do you see within the block? Within the block, do you see T3, V3 anywhere else within the block?
No, sir.
No. So this has to be replicated. So that is why we have block 2. So block 2, you see where is T3, V3? This plot is T3, V3. You see, it is replicated 2 times. So 2 times mean block 1, block 2. These blocks are actually functioning as replicates, right? These are functioning as replicates. Okay. Any questions on this? So that is why we have blocks. Blocks is for, this is actually replication, right? That is why we have blocks. Any questions? This is the problem where I said that here in this case, if I say factor, here we have tillage, 3 factors, right? Tillage, that is main effects. The other one is varieties. The third one, tillage into varieties. So these 2 are main factor. This is interaction. Do you remember we talked about factorial? So here, when you talk about this for our precision, in terms of precision as well as for estimations of these values, right? Similar to our nested design, here also our denominators are different. Our denominators are different. For example, tillage practice, is it in main plot or subplot? Here, tillage is coming in main plot or subplot? Here we have main plot. I will detach MP. Varieties? Subplot. What about tillage into variety?
Subplot.
Subplot. You see, these 2 are in subplot. This is in main plot. So these 2 will be treated similarly. This will be treated different, right? We will come to that in a little while. This main plot analysis treated separately or differently compared to the subplots. The reason is they are producing different levels of precisions, right? Therefore, the errors are also differs. When you say errors, it is the calculated value. Error values are needed here as the denominator. So the experimental errors are, especially, what is the purpose of experimental error? To test with to find out the ratio with the treatment. That is why we need experimental error. So here we are going to use different experimental errors. Suppose if you have, I said here, main plot and subplot. So we have 2 errors. So error 1, for example, error 2. If you have 2 plot size, we have 2 errors. If you have 3 plot size, 3 errors. I will talk about 3 plots later. First, we will understand only 2 plots, right? Right. Speaking about that, we can say, because there are 2 size of plots in this case, main plot and the subplot. Therefore, we have 2 experimental errors for each plot size. One is for main plot, the other one is for subplot. 2 plot size, therefore 2 errors. Usually, subplot error is smaller and have more degrees of freedom. Subplots usually have smaller size, and also degrees of freedom is quite high. We will see that later. Therefore, the main plot factor is estimated with less precision than the subplot and interaction effect. So, can you remember main plot? Tillage is main plot. The varieties as well as the interaction between tillage and variety are in the subplot. So, subplot are treated differently from main plot. So, main plot is having less precision compared to the subplot, right? So, that is the concept here. Precision is an important consideration in deciding which factor we should assign as the main plot. So, we cannot get the precision equally for factor A and factor B and the interaction. Interaction, not a problem. It is always in the subplot, but we'll have to decide which has to become as a main plot, whether it is factor A or factor B. There are some advantages of this split plot. It permits the efficient use of some factors that require different size of plots for their application. So this is what we need. For example, we said tillage. Let's say pesticide application. You are using chemical pesticide, one treatment. Can you spray pesticide within a small area? What will happen if you spray 2 into 2 square meter plot? What will happen? That will spread to the other field also. Because if it is if there's an airflow, if you spray it will go. So you see, so that need is a fairly large plot. So likewise, we can talk about further. The other advantage is it permits the introduction of new treatments into an experiment that is already in progress. That is another important aspect. What do you mean by new experiment? For example, let's say I have started one experiment. I have started one experiment. Let's say I have factor A, A1, A2, A3, B1, B2, B3. Now, suddenly after 2 weeks or after 3 weeks of establishing, this is, let's say B is variety, A is planting dates, now I want to introduce a new factor C, that is feeding. Right? So in the weeding, we could say it's 3 types: manual weeding, weedicide, and mechanical. Manual, using chemical, right? Manual, chemical, and mechanical. Let's say we have 3 types of weed control. So weed control is coming maybe after establishment only we think it. We want to apply. Now I can apply this like this. I'm just showing in one plot. I'm showing in one plot. You can do it in others. So let's say this is C1, C2, C3. So this kind of, how many size of plots are there here? How many plots are there in this example?
3 size of
Three sides. So we could say A is in main plot, B is in subplot, C in subplot. You see, this is we introduced because initially we didn't thought about it having as the experiment. So we got it using some knowledge. We got it later on. We thought about it. We want to introduce the third factor. So that is why we have already introduced that. Okay. So here, this is one way. This is main plot. B is a subplot. C is subplot. Smallest plot. So how many plots are here? 3 types of plots. So how many types of error there will be? Experimental error? 3 types.
3 times.
E1, E2, E3, because we have 3 plot size. Remember that. Right? Okay. So likewise, this is one step further, third one, right? Split plot. All right. So this is the second type, which I discussed with you all just now. It's called as, this is called as split plot. Split plot. Why? It's dividing one plot 2 times. Right. So this is the main plot divided into first time into B1, B2, B3. That is the first dividing. Second dividing is C1, C2, C3. So 2 times it has been splitted. So it is called as split plot design. So this is a bit complicated. I mean, third level is more complicated, but still people are doing. So the more convenient one is 2 levels of dividing, which is called as split plot design. Right. So these are the advantage. Hopefully you have already written down the advantage. Okay. Disadvantages. There are some disadvantages also. Already one disadvantage we have discussed. What is that? The precision. Precision will be different. Right? Precision will be different. For example, here, look at it in this case. We already said here 3 factors involved. Tell me all the interaction factor A, factor B, factor C, AB, AC, BC, ABC. Can you tell me which plot size according to this? What are the plot size? A is in early example. A is in main plot or subplot or subplot?
Main plot.
Main plot. What about B?
Subplots.
Subplot. What about C?
Subplot.
Fill the rest. I will give you 30 seconds, fill the rest. Right, now tell me, what about AB?
That was a subplot.
That should be,
Subplot.
okay. What about AC?
AC, BC also. AC is subplots.
Okay. What about BC?
BC also subplot.
ABC?
That is subplot.
You see, when you estimate these values, you see, this is high precision. This is plus this. Then what is the next precision? Next highest precision? AB and B. So 3 precision level. This is the last one. This is the low precision. So we have 3 precision level. We can evaluate all these interactions, but you see the precision level are different. So these 4 with high precision, 2 with relatively low precision, and 1 with lowest precision. You see, this is the disadvantage of split plot design. So here, this is what it has been explained. 2 disadvantage. One is the main plot is estimated with less precision, so larger differences are required for significance because the error term is very large. So that's why we tell the larger differences is needed. Maybe difficult to obtain adequate degrees of freedom. That is also one of the problems in the main plot. The second one is it's more complicated. We will do using by SAS. It is very, very complicated because the programming is difficult. The rest is easy. Yesterday we learned about the last part, programming. If you miss one of this, it will be difficult because it's easy for do it by using software, right? So it's difficult, but anyhow we will learn the commands, right? So that is the disadvantage. Data analysis. This is the form of factorial experiment, so that analysis is handled in a similar way. No difference between the factorial and here in the analysis, right? Especially in the calculation. We will estimate the test, the appropriate main effects and the interaction. So that the analysis procedures as follows. Make a note of it. This is the analysis procedure. First, you construct a table of means if necessary. Otherwise, not necessary to follow the same structure, but we can complete an analysis of variance. That is the Anova table. Perform the significant test that we will do it. Then the computing means and will be done by computer once we see the computer later on, computer application later on, and you'll have to interpret the analysis. This is the Anova that we can come up with. I will just brief it. I'm not sure whether I have. No, this is not necessary. Just a second. Right. Take down this the summary of the analysis of variance. This is for split plot design, not split plot design. This is for split plot design. Analysis of variance, right? Take a note of it, then we will come up with the notations in a few seconds. And the model as well. We'll come up with the model then Anova table. This is the summary Anova table for a two-factor case. For a two-factor case. So we have main plot and the subplot and the blocks. Have you all written down? This is the summary. I'm going to give the notations in now. In the slide, notations are not there. So here we have two factors. So that two plot size. So because of two plot size, you see we have two errors, error A and error B. So we are using error A to test only the main plot. We are using error B to test the main subplot of factor B and the interaction effect. If you have written down, tell me so that I can give the model and the sums of squares. Oh, Tanesh Kumar?
Yes, sir.
ക്ലാസ്സുകൾ ഏതെങ്കിലും പോ
ಇಲ್ಲ ಇಲ್ಲ
റൈറ്റ് ഇല്ല ഞാൻ கொஞ்சம் ஒரு 5 நிமிஷம் அப்படி கூட எடுத்தாலும்
ಓಕೆ ಸರ್ ಓಕೆ
I'm not sure, but we will see. I'm searching for an example. Have you all written down? Anyone? If they have completed, just tell me. I'm looking for an example. Hello. எல்லாம் தெரியும்
ಇಲ್ಲ ಸರ್ ಓಕೆ ಸರ್
சொன்னா தானே நான் மெட்ட வேலைக்கு போகலாம். Right, so we will write down the Anova table. Before that, the model, let's say first we will say the model for split plot. Model for split plot design. Right? Model. So when you say split plot design, how many factors are involved? When I say split plot design, how many factors are involved?
2 factors.
Two factors. So A and B factor. two factors are involved, right? So let's say y I j k is equal grand mean plus we could say alpha I, doesn't matter whatever the we will take first I will take alpha beta, yes, tau I plus the alpha I j plus w I j plus a bit complicated this, as we said, compared to the earlier designs. I will explain all this. Then we have beta k plus alpha beta jk plus the error term eijk. So this much of components are coming into picture. We can explain all these components. So this is, can somebody tell me what is this yijk, how we can define? Observed value of Peony. Tell me what is a yijk, observed value of. So before we say what is the we define the ijk. Yes, give me a second. We will define what is I. I is from 1 to up to R. It is the blocks, number of blocks. J is starting from 1 up to A. It is the levels of A. K is the 1 to up to B. The levels of B. Right? Right. Now you can define what is yijk. What is yijk?
J level of factor A.
Yeah. We'll start from I. It is the observed value.
Observed
in the
value in the
ith block.
ith block in the level of A and level of B.
No, ith block. Then before talking about level of A, jth level of A. Jth level of. So we could say observed value of ith block, jth level of factor A, and kth level of factor B. You can write down. You'll have to include all this: kth level of factor A, sorry, jth level of factor A, kth level of factor B, and so on. Right. What is mu? Everybody knows grand mean. What is tau I then? I have already defined what is I. What is tau I?
Effect of the block.
Yeah, it is ith block of ith effect of ith block. What about wij then? Actually, it is called as error A. Also, it can be said as it is the interaction between ith block and jth level of A. Actually, it is the interaction effect. That is what we are considering it as A, error A. Before that, we missed one thing. Before that, we missed one thing. I can remember now. Yes. What is that? In the model, I missed one.
Alpha J.
Alpha J. So you write alpha J first. What is alpha J? It is the effect of
Factor A.
jth level of
Jth level of factor.
factor A. Right. After that, you write the next one. Then what is next? Beta. Okay. It's simple. Effect of kth level of factor B. Then what is alpha beta? We have already seen this, even in factorial. It is the interaction effect J. No, it is not IJ, right? It is JK. Jth level of factor A and kth level of factor B. Right? What is the last? Error term, YJK. You can explain. It is the random error of random error. If you want, you can say it is the error term or random effect. We call it as random effect of ith block, jth level of factor A and kth level of factor B. Write down very nicely. I have simply written in a short info. Write down nicely. Right. Any questions? This is the model.
What is WIS?
That is the error term, which is giving us the interaction between the block and the factor A. So I have just simply put a notation. You can put any notation. Xij. Doesn't matter. இது பைபிள் இல்லையா? இது பகவத்கீதை மிக குர்ஆன் ஓ மகாவன்சனமே. So this you can write any notation. This is just to indicate the notation. You can use any notations, right? I have used Wij to indicate that is an error term, right? So you see in this Anova table, how many components are coming? 1, 2, 3, 4, 5, 6, right? 6 components are coming. We'll have to describe all these 6 components in the Anova table. So here we have described what is this block effect. In our model, how did we indicate this as? tau I. What is the effect of A? Look at the model.
Alpha J.
Alpha J.
J.
What is error A?
WIJ.
B?
Beta K.
Yes. This is alpha beta JK. And this is the error term IJK. You see, all the components are there. We know mu is a grand mean. Mu is a grand mean. All the components are there. Is it clear? Pamelaita, Tanis?
Yes, sir.
Right. We will continue with the Anova table. So the same setup is coming in the Anova table, right? Don't be surprised by looking at this Anova table. So Anova table for the above model. So I will very briefly say is the source of variance, degrees of freedom, sums of square. Yes, we will go for the sums of square, then we will look at the mean square. Right, so as you all know, first one is block. Degrees of freedom is r minus 1, already I have defined. So summation y I dot, sorry, only 2 dot square divided by AB minus the correction factor. What is correction factor? Y dot whole thing square divided by here R AB. That is my correction factor. So hereafter, if I say in this example C, that is the same. Right. Write down for factor A minus 1 is the degrees of freedom. Summation Y dot J dot square divided by R B minus correction factor. RB, or you can write BR, whatever it is. Because since it's indicating the first dot as R, so that is why I'm saying RB, so that you can understand. The next one, this is about the main plot. Before going to the subplot, we'll have to give the error. So I will say error A. So what is this error A is actually, it is the interaction between block and factor A. So I write it in simple term. It is the interaction between block A and, interaction between block and factor A. So you also can write. So you can think about how we can formulate your sums of square. So that this will be given as R minus 1 into A minus 1 in the degrees of freedom. Can you remember the interaction effect? How did we calculate for AB interaction in the factorial? Same thing. So there is summation Y, right? IJ, interaction effect between block and A, not B, square, divided by A, sorry, not A, B, minus C, minus block sums of square minus sums of square due to A. Block sums of square minus sums of square due to A. So which we have already calculated in block sums of square and the A sums of square. Then we go for the next one, factor B. So the degrees of freedom is obviously b minus 1. The sum square is also easy. That is y dot k square divided by ra minus c. Then we come to the interaction between a and b. So that is degrees of freedom a minus 1 into b minus 1, right? Then the normal interaction, as we said, summation y dot j k square divided by r minus c minus sums square due to a minus sums square due to b. Then the last one, the total, then we can calculate the error. Total degrees of freedom is ABR minus 1. Correct? As usual, total YIJK square minus C. Then the error B can be calculated, total minus rest, so that A into R minus 1 into B minus 1. So this we can write it like that, the error degrees of freedom. Then we can say total sums square minus sums square due to A minus sums square due to B minus sums square due to AB minus sums square due to error A minus one more sums square, that is block sums square. So you see, you divide, you get rid of all. Right? Okay. Now, mean square is simple. Mean square is, as you all know, mean square is, we can calculate the mean square, not a problem. Mean square for, usually mean square not necessary for block, but mean square for, I will put it as mean square for A, right? You can calculate the rest of the mean square. Mean square for error A, that also you can calculate the mean square. Mean square for B, we can calculate. Simply we divide by the degrees of freedom. Right? Mean square for AB, and we can get the mean square for error B. So here we have two errors, so that you separately calculate the mean square for these two errors separately. Then, as usual, we can test it. The testing, can you tell me if I want to test all these components? So let's say if I want to test A, what is the mean square that I will use? Effect of A, main effect of A. What is the mean square that I will use as numerator and the denominator? So as you all know, my null hypothesis, according to this, is alpha I is equal 0. That is my null hypothesis. Tell me what is the numerator and the denominator in testing. Tell in mean squares. Mean square of A divided by what mean square?
Means of error A.
Yes, mean square of error. Right? Now, if you go to main effect of B, what is the F calculated value? Mean square of B divided by mean square of error B. Right? So what is here my null hypothesis? Beta j is equal 0. Then the third one, the interaction effect of AB. So the calculated value is mean square of interaction AB divided by mean square of error B. Where here we testing the null hypothesis as alpha beta ij. Sorry, not ij. Here some mistake. Here we are not k. Sorry, jk. Careful about your. This is also there's a mistake. Because we are not taking I as the block. So that alpha j, beta k. Alpha j, beta k. Right? So we can test all this and then look at their degrees of freedom of numerator and denominator, then we can compare. Right, we have a few homeworks. First homework is write down the model and ANOVA for split plot design. So that means it has, as I pointed out, it has 3 size of plots. 3 size of plots. For example, look at it. So we have the black color plots are 1 plot, blue colors are, I will say, this is 1. So this is 1 plot size, very small. It is the subplot. The other plot size is second one. It is the medium size. It is called a subplot. The third plot size is for example, this one. I have used different color, doesn't matter. So the blue color is main plot, black color is subplot, the pink color is subplot. So this is called as split plot design. 3. So we'll have to give the model and the ANOVA. Right? Now I will get you example. Write down this example. First you write down, then you can draw it. A field experiment was conducted. Write down. A field experiment was conducted to study the effect of irrigation and nitrogen. The field experiment was conducted to study the effect of irrigation and nitrogen on paddy yield. Right? Is it clear? The design used was split plot design with 3 blocks. Split plot design in 3 blocks, with 3 blocks. I will give the summary of the data. That would be easy. Right? 2 levels for irrigation. 2 levels of irrigation. Two levels of irrigation. Within the bracket, under irrigated water and within the bracket under irrigated water and under rain fed. Right? And 3 levels of nitrogen and 3 level of nitrogen within the bracket 0, 15 kilogram per hectare and 30 kilogram per hectare were used in the experiment. Right? The results recorded from the experiments, are given below. The results recorded from the experiments are given below. Write down this table. Write down this table. Right? Test for main effects and the interaction effect, and test for main effects and the interaction effects, and express your conclusion. Express your conclusion with alpha 0.05. Express your conclusion test and express your conclusion with alpha 0.05. Right. So you can take this as a homework. We can see that probably next time. Right. Any questions? So here, irrigation level, there are two levels, I1, I2. This is the main plot. Then we have nitrogen, we have 3, N1, N2, N3. So this is the subplot, is the main plot. So that is the experiment. So we have given 3 plots. That is fine. Any questions? If not, I think we will wind up and see you in the afternoon. Right. Okay. Bye.
Sir, excuse me.
Thank you, sir.
Okay. Bye.